Today's conversation is with Dr. Nadanai Laohakunakorn. Nadanai is the leader of the Synthetic Biophysical Systems Group at the University of Edinburgh, where they research cell-free synthetic biology, bioengineering, gene expression biophysics, and microfluidics. This research has applications in biosensing and healthcare and provides new understanding of life by recreating its processes from the bottom up. It was exciting to talk in detail about the science, from protein synthesis to nanopore DNA sequencing. It was also equally informative to talk about some of the practical processes that all scientists must experience, like having their ideas for research scrutinized before receiving funding. Overall, it was simply a fun conversation. Here is our conversation with Nadanai Laohakunakorn.
Yeah. So, Nadanai, please, can you please tell us about your career so far and what have you enjoyed about it being a scientist, and what are some projects that you found interesting that you like to tell us about?
Yeah, sure. So I started off studying natural sciences at Cambridge, which is one of this, it's a sort of broad degree where you start off studying all the sciences in the first year, and then you eventually specialize. And I specialized in physics. And my PhD, I actually remained in Cambridge doing my PhD, where we looked at physics of nanopores. So nanopores are these sort of tiny openings in either membranes or sort of a solid state substrate. And what we used them for was for sensing. So when you drive an ionic current, so the nanopore joins two chambers which are filled with a salt solution, and you can drive an ionic current through the nanopore. And these nanopores are very small, so single molecules can pass through them. And when the molecules pass, they modulate the ionic current. And so you see a change in your signal, and hence you've detected the molecule. And the dream at that time, so I started my PhD in 2011. The dream at that time was that you could pull a DNA molecule through a nanopore. And based on the structure of the DNA molecule, the different bases, you get different modulations in the current. And hence, if you pull the DNA very carefully through the pore, you could sequence the DNA using a physical method. At that time, it was not yet possible, but now it's actually an established technology. So there's a company called Oxford Nanopore Technologies, and they use these small nanopore devices to sequence DNA very quickly, which is really amazing. At that time, we didn't think it was possible. So my research was to do with the physics, and more specifically the fluid flows at really small scales when DNA molecules move through these pores. But I basically specialized. I was actually interested in biophysics because my degree was sort of very broad, and I studied biology as well as physics. But in the PhD, I kind of ended up specializing in fluid flows and nanopores, which is very sort of obviously very specialized. But then for my postdoc after my PhD, I wanted to move more into something biological. So I used my knowledge of microfluidics and nanofluidics, these fluid flows at these very small scales, and applied that. So I moved to Lausanne in Switzerland to work on what's called cell-free synthetic biology. So this is where you take components from cells, which are active in vitro, active outside the cell, and you recapitulate biological processes using these parts. So it's a non-living reaction, but it's doing something which the cell does. So in particular, what we looked at was gene expression, so transcription and translation in vitro. And we used microfluidic reactors to control these chemical reactions. So you could accurately introduce the various components and change the conditions. And so you have some sort of almost like a simulation, a physical simulation of a biological process on a microfluidic device. So I was in Lausanne for nearly 4 years, and then I moved to Edinburgh to start my own group, and we're carrying on with this in this line of research. So I'm sorry, I forgot your question. So that was my background, but you asked me about something about my experience.
That is the core of the question to
Okay.
gain a bit of knowledge about your background
Sure.
and your
Yeah.
experiences in the science field. So
Yeah.
yeah, personally, as I told you, I'm very interested in physics, and Carlo and Yash are personally very interested in biology.
Yeah.
And we're really interested, how is it like to combine biology and physics and biophysics? Because I personally don't have a lot of knowledge about it, and it sounds very exciting.
So the thing is, both fields. I think the main difficulty is that both fields have a very strong culture. So when you study physics, for instance, you learn, everyone studying physics around the world learns the same things, the same concepts, the same sort of theories, if you like, and you learn it in a certain way, and you learn a certain way of thinking, which is accepted worldwide. It's the sort of physics way, if you like. If you learn biology, it's sort of similar, but your way of thinking and the things you learn obviously are completely different. I think in the end, the main difference, I mean, obviously the material is different, but the way of thinking is also slightly different. So if you decide to do biophysics, you kind of have a choice, whether to, let's say, adopt a really physical way of thinking, or try to adopt more sort of biological concepts. This is the really interesting part of it, I think, and it's also the main challenge, is which bits do you use from the different subjects to think about your own research.
Right, very interesting. And speaking of that, actually, being the cultures being very dominant, how did you come to choose science, and specifically biophysics, in your career?
Well, I mean, I was always interested in science. I think ever since I was young, and so it was a natural choice for me to study science. But I was not set on a research career at all. This came later, so I didn't. I studied science because I found it interesting and I wanted to know more about it. And I enjoyed, in my undergraduate, having all these different sciences. So I studied physics, I studied chemistry, I studied biology at first year, at least first and second year level undergraduate. And I saw how different they were, but also how much actually, like, fundamentally, there's a lot of things in common between all these disciplines, right? Because in the end, these boundaries that we impose are purely artificial, because you can look at whatever process you like, whatever natural process, using all sorts of different approaches. And I think that's what attracted me to this interdisciplinary approach.
Mm-hmm. Right. I guess each person defines success in their career in a different way. What are some key steps you think students like us, getting to undergraduate courses, can take and to establish a successful and fruitful career, basically, later on?
Yeah, I think this is something that my opinion on this has changed as I've progressed in my career, and nowadays I would say that so much of the success, so much of your satisfaction with the career is personal, is internal. So that really the most important thing that I would do if I were in your shoes is to find your motivation, to find really think about why you want to do these, why you want to do this career, and keep that sort of at the back of your mind the whole time. If you're motivated by an external success, so let's say if you want to do a career because you want to be famous or you want to win an award or something, I don't think that's a really good reason for doing a career, because chances are you might not become famous or win an award. And does that mean your career is not successful? I don't think so. But if your reasons for doing a career are because, you find something interesting, you're curious to find out more about it, you want to learn and you want to teach people, you can have a really successful career. It's your sort of point of view, right? And if you're having a really good time with your career and doing all these things that you love, and you happen to win awards and gain recognition, well, that's a consequence of your kind of personal success in the career. So I think if you look at it in that way, this is just my take on it, because obviously, there are people, and I used to be a bit like this as well. I think when I was younger, when I thought, well, you have to win a big award, you have to publish a big paper, and that's what you're chasing. But actually, in the end, those are consequences of a career where you can be personally satisfied.
Yeah, that seems like a very common theme between all the scientists that we've talked to actually as well.
Yeah.
It seems like it's a personal motivation aspect, and it's on that note, it is also utilitarian in terms of their science as well. So it's passion and utility at the same time. So
Yeah, I mean, it's for me, this is kind of fundamental to everything. Let's say if you actually just look at something specific, like a research project, you want to do a research project. Obviously, you want to, you propose a hypothesis, you want to see some results, and this motivates you to go into the lab to do the experiments, to do the hard work to do this project. If you don't enjoy being in the lab, if you don't enjoy actually doing the work, you'll have a miserable time, because if you're motivated by the final result, but not by the process, then you spend, basically 90% of your time not being really happy until you get the final result. But obviously, if you're not having a good time in the lab, the chances are you might never get to the final result, right? Whereas actually, if your reason for doing the project is you actually love being in the lab and, sort of looking at these sort of interesting things going on, and the final result happens to come out as a consequence, then you spend sort of all of your time being quite happy in the lab. People will say, you spend so much time in the lab, are you not sort of, you're working too hard? But if the work is fun, it's not really work in a way. So you're just having a good time, and then, the results come as a consequence of all of this. So it's a sort of different way of thinking about it, I think.
A lot of the people who talk to, they've when they recount like their experience in science, a lot of them talk about travel. When they were studying between their PhDs and postdoc work. How common do you think that is, and do you think it's a necessity?
I mean, I don't think it's a necessity. You can probably get away with not traveling, but I think it would be a great shame because, well, first of all, when you travel, you just expand your horizons. You just meet so many different people and you see different ways of doing things. Even in the same country, among different universities, different institutions, there are different ways of doing things. And there's no one correct way. So, the more you see, the more you can take sort of the good aspects, if you like, from the different things you see and create your own sort of working environment, or your own approach to something. So that's one thing. I think, sort of discovering different ways of working, which are very, very different in different places. And obviously the other thing is you meet people, because great people sort of, or, really interesting, really intelligent, really sort of people you'd like to know, let's say, they exist everywhere. So they're not confined to one place. So the more you get to go around, the more you're able to meet these amazing people, and then potentially you could work together, because really science it's an international effort, right? There's no one country which owns science, so you want to work with the best people wherever they are.
So is that international collaboration? Is that becoming more common? It's like how often do you say are you working on projects that involve like members from different places of the world? Like how does that work?
In my experience, I've always worked with international collaborators, so there's never been any point in my career where I haven't. Is it more common? I'm not sure. I guess I've not been around long enough to know how it's changed. But obviously, online sort of collaborations, or even just collaborations using Zoom and things like that, but also sort of online platforms for writing papers or sharing results is now easier than ever. So I imagine it's the easiest it's ever been right now to collaborate.
Sure.
Because you hear stories. I've not, as I said, I've not been around long enough, but the more senior professors in my institute will tell me about the times when in order to collaborate on a writing a paper, you have to basically type it up on a typewriter and post it to someone, and they'll correct it and post it back to you, right? Obviously we can't imagine doing that today, but that was how it was done, I don't know, 30, 40 years ago.
Sure. And so as we were reading some of your work, she was really interested in some of the work that you're researching now. And so we'd just like you to talk a bit more about the research you're doing in your lab. Yeah,
so there are lots of things we're doing in the lab at the moment. So my lab is obviously new. So we started only 2 years ago. And with the situation in the world being as it is, it's been a big struggle to get the lab up and running. So it was a difficult couple of years. But now we're sort of we're making a little bit more progress. We don't have any sort of really new things yet. At the moment, the first thing you do in a new lab is you try to set everything up and sort of reproduce all the old stuff that you were doing before, to make sure your lab is sort of working as it should before you try something really, really new. There are a number of questions we're studying in the lab. So one of the main questions I'm interested in is something called self-regeneration. So it's a process by which, all cells have to regenerate their internal components, right? And this is and eventually this allows them to grow and divide if they're growing cells, but it also allows them to maintain themselves if they're not growing, because obviously they have to repair and turn over their various internal components which eventually degrade. So I'm interested in regeneration because I think it's one of these sort of hallmarks. It's one of these hallmarks of life. It's a key process which all living things have to do. And I guess people don't study it so much sort of by itself. It's usually part of something else. If you're studying cell growth, or if you're studying homeostasis or something, it's sort of there in the background, but you never sort of isolate it. What we're doing is we're using cell-free protein synthesis systems. So these are, as I mentioned earlier, these are biochemical reactions where you have components from the cell. So protein components like ribosomes, polymerases, various enzymes which you need for gene expression. And you have small molecule components, so substrates for these, such as NTPs, amino acids. And you mix them together, and typically what you do is you use the reaction to make some protein. But if we're interested in regeneration, what we can do is we can take the reaction and use it to make more of the components inside the reaction, if you see what I mean. So you might choose to make more RNA polymerase, or you can choose to make more synthetase, which is an enzyme which charges tRNAs before they're used to engage in polypeptide synthesis. So in my postdoc, we studied doing a little bit of this, using the reaction to make more of its own components. Eventually, a very hard thing, but one of the goals of the field is to use the reaction to make all of its protein components, right? Because if the reaction can do that, you potentially could encapsulate it in a synthetic cell, and it could drive the growth and eventually replication of synthetic cells. So it's a way of trying to recreate some process that we see in biology, but actually from what's called the bottom up. So actually you use the parts. And this is a really powerful approach because once you start trying to do this in the lab, you see how difficult it is. You see that the regenerative process is prone to failure. It's kind of very fragile. Biology obviously can do it very robustly and reliably. So what's the secret, right? That's the big question. How can biology do this so well, and what don't we know, right? What are we missing from our system? So this is the main sort of, I guess, scientific question that we're tackling in the lab, and it has interesting applications, as I said, for the construction of synthetic cells, for instance. But also fundamentally, I think it's a very interesting question.
And is this a new field? Like, is it difficult, like, convincing people like the importance of the problems or the questions that you're trying to answer? Like, what's that like?
The field itself is, I guess it's kind of new. So synthetic biology has been around historically synthetic biology has been around for a long time. But the synthetic biology as we know it, when you mention synthetic biology to people, typically what they have in mind is probably the last 20 years of development in the field. And this is basically due to the fact that we are now able to synthesize DNA very cheaply. So we're also able to edit DNA, assemble DNA very well, very easily. So these are these new tools which have arisen in the last 20 years or so. So it means we're able to make very precise changes to cells. And at the same time, there was this development of this cell-free approach, which I've been talking about, where you actually just use this DNA and components to make in vitro reactions, which you can program with essentially genetic material, arbitrary genetic material. You can just synthesize whatever DNA you want, put it in a test tube, and then the test tube will operate this sort of genetic program for you. So this kind of approach, the approach itself has been around for a very long time. So cell-free protein synthesis arose when people were trying to figure out how protein synthesis worked. So back in the 50s and 60s. But today we have this huge benefit of DNA technology, which allows us to really harness it in a powerful way. And so in that sense, the field itself is it's developing rapidly. There are many people in the field doing really interesting things, there are all sorts of applications, but also fundamental questions being asked. In terms of regeneration, again, this question has been around for a long time, but it's a little bit more sort of niche, let's say. There are not so many people working on it. So it's kind of an effort for us to sort of carve out our own space in the field. Obviously, we depend on a lot of other results in the field, which help support what we want to do. in terms of convincing people that it's important, I don't know. So we've been quite successful with getting funding for our projects. So it seems to be okay for now. It's not so obscure that people are not interested in it. But it's also kind of not so common that people think it's sort of boring.
I wouldn't call it boring. You're just describing it. It's making me really excited. Now I just want to go read about it. It's awesome.
Well, no, I mean, boring is a very subjective word, right? But I think, so when you propose, if you want to propose a research topic, later on in your careers, when you start developing your own projects, you'll have to apply for, funding to help support the project. And then, choosing how you want to pitch your project to potential funders, like that's a key sort of career skill for scientists, I think.
Sure.
So you have to pitch it at exactly the right level. So as I said, the project can't be too easy or too common. Not too many. You don't want lots of people to sort of. You don't want to choose something that a lot of people have already worked on. But at the same time, it can't be too obscure, because if it's too obscure, then people will think, well, is this really a thing? Yeah.
Sure. And also, I'm also just curious now. Is it how do you actually start a lab, and how common is it? How common is it? What are the prerequisites? How do you sort of convince people? I suppose that. Or I suppose what makes you a better person to run a lab than say some other person who wants to do it? How does that work? How's it allocated?
What makes you a better person? That's a really sort of deep and difficult question.
Sorry, I think I phrased that wrong. Yeah,
no.
It's person, but it's candidates. Because imagine there's a limited
of course. No.
So how does it get selected?
The practical answer is sort of easier. So typically it's basically like a job application, right? So if you have your PhD, let's say, and you have some experience working as a postdoc, and you feel you're ready to apply for the job, it's essentially a job application. So you apply to research institutes or universities, and it's a competitive application. So this is where the interview panel will assess quite rigorously all the different applicants. The process is different in different countries, but essentially it involves an interview. It involves looking at your track record, and potentially meeting with a lot of people in the new university to see what. So you kind of get assessed by an interview panel, but also informally assessed by your future colleagues. And there are many things they take into consideration for deciding whether or not to appoint you, right? Which is, and this is a really difficult process, because essentially you want someone who's able to lead, but obviously if they haven't led a lab before, they don't know, they don't have a track record, so you kind of have to assess their potential. And then you also want someone who's sort of strategically good for the department. So potentially, if they work on something which a lot of people are interested in your future department, then that's a big benefit. And eventually, so they may decide to make you an offer. And typically, this involves them giving you, a little bit of lab space and maybe a little bit of money to help start your lab, and then basically you join the university and you start trying to bring in more money to launch your research projects.
Sure.
Yeah.
Actually, we're also just curious about the day-to-day life of a scientist. It's what does a day in your lab look like? What is it for you?
Well, in the morning I go on a podcast to talk to people. No, it's quite variable. This is something I did not know so well before I became a scientist. Because when you're a PhD or a postdoc, your day-to-day life is just your research project. You go into the lab and you do things, and eventually you sort of write everything up and publish it. as a scientist, you oversee these things going on in the lab. So I have four PhD students under my supervision. So I try to meet with them every week to discuss how their projects are. So I don't actually do the projects for them, but I sort of oversee the projects and I help guide them if they get stuck, or if they're not sure where to go next. I sort of give them ideas. But essentially, it's quite a high level thing I do. So I guide them broadly where to go, and typically they solve the technical problems by themselves, because in the end, the technical expertise you need to do a project is quite specific to that project. So I have technical expertise in a number of areas which may not be related exactly to the projects which are going on in my lab. So I can't help them so much there, right? So it's this kind of slightly higher level management of the project, where you're helping just steer them, but you're not doing the actual project for them. So essentially I oversee, I meet with my students, I meet with my collaborators. There's typically a number of things you have to do sort of administration-wise for the university. So the various teaching responsibilities I have, and meeting in various committees. So there's a lot of meetings and stuff, which is kind of not really science, but it's there and you have to do it. So I find it really, really important to set aside some time to actually sort of read and think about science. And this is something I would say it's usually the thing which, well, at least in my experience, it's the thing which has been sort of encroached upon the fastest. So as soon as you start working, people ask you to do a number of things which are not really related to your research. And you have to try to protect some time to think about your research. At the moment, I also spend some time in the lab, and I'm quite keen to do that, because I still train. We have a new PhD student. I'm training her, so I actually go to the lab a couple of days a week to train her to do something specific in the lab. Possibly in the future, I will do less of that. At the moment, at the beginning of your career, you still have a lot of sort of expertise in the lab. And for me, it was important to pass that on to the students as quickly as possible, also before I forget it, because if you don't spend enough, if you leave the lab for a long time, you won't be so up to date anymore with how to do things. So I still try to do some lab work, but it's really not. But most of my time is actually not. It's actually meeting with people, meeting with my group, and this actually was something. As I said, it was something which I didn't realize would be the case so much before I started. I thought I would have more time to think about science, essentially.
Do you think that's a common experience amongst people who leave labs? Is just not enough time to be able to do the science itself? Is there too much responsibility? Is that common?
I think so. At least within my sort of experience of chatting to colleagues, I think this is a common problem with everyone. So I think basically what you have to do is you really have to try to protect some time for yourself, because I guess it's similar to any sort of management role, any role where you have some responsibilities. You end up with a lot of things to take care of, and if you're sort of too nice, or if you end up saying yes to everything, then you actually sort of fill up your calendar with things which are not really related, not really what you actually want to do, right? So it's
Sure. Now, the next question's a bit broader. It's about academia as a whole. Academia, I think we've heard it described as a place where ideas are like rigorously scrutinized, and they go through a sort of battlefield But do you think that gets in the way of exploring like novel ideas that might be out of the box? Is that, or do you have any experience with that?
That's a really interesting question. Um, I have to think a little bit about this. I don't
Sure.
have so much experience with this. I mean, I would say, I mean, in my limited experience, the big projects which I have funded, which have been given funding in my lab, are have been all quite rigorously scrutinized. So if you propose something that you want to do, and you want to get money, or sometimes significant money to do that project, it has to go through a process of peer review, where people will look at the project in quite a lot of detail and see whether it's feasible to do or not, right? So, and I think that is actually important, because it helps prevent people from sort of basically funding really bad research, right? So you do have to have some scrutiny there. But then in my lab, we actually have a lot of kind of really crazy things we try to do. I mean, not even crazy. Like sometimes you just want to see what happens when you do this, right? And you don't have money to do that, and probably no one's going to fund you to do that. But you basically choose something really small to test. And this I use basically from, basically I designed these very small projects for undergraduates. So we have undergraduate students who do their final year projects in the lab. And we also have master students who do short projects in the lab. And these projects, for me, they're kind of ideal to test out these slightly wacky ideas, which may not work at all, right? So, I mean, I guess, I mean, obviously the students know this when they start. Like, I don't tell them, like, let's do this project. It might not work. I mean, there's always a chance of projects, even the really well-funded ones, not working. So that's always a part of research. But yeah, so the really novel things. So typically, what you might do is you might have some crazy idea, which you have no idea whether it's going to work, but actually it's quite easy to try out. So you try something with maybe an undergraduate or a master's student in the lab, and you see what you see. And sometimes there might be an interesting result. If there's an interesting result there, you can probe it further, right? You can see if it's a real effect or not. And if it looks like it might be a real effect, then you have a little bit of preliminary data already, and you can use that to apply for a bigger project. So there is a kind of vetting process of ideas. So if you have a really crazy idea, you have to kind of test it out by yourself first.
Sure.
And I think most people do this, right? This is how new ideas happen. And then by the time it gets to be, fully vetted, and official money comes in to help support it, it's already kind of established, more established. So I don't think, yeah. So I guess in answer to your question, if you have a really new idea and you have an easy way of trying to test it out, then it's possible to do. If it's a new idea and to test it is really, really expensive, that probably won't be possible. So in that case, typically, you have to find another strategy. It could be that, you have to find money from elsewhere. It might not be a normal sort of research grant.
Sure.
Yeah, that's right. And following up on that note of, scientific ideas and its filtrations, what do you think are some other unpleasant aspects of being a scientist, and how can one avoid them, basically?
Probably there are two things I think probably, which I didn't know so much before I became a scientist. I was not like you guys. I was not so. I did not know so much about the career. So the two things revolve around uncertainty, which can be unpleasant. So first is uncertainty in funding. A lot of scientists might say that this is like the worst part of being a scientist, is you always have to ask for money to do something, right? And the thing with applying for money is that a lot of the time you won't get it. It'll be rejected just because, statistically speaking, sort of research grants have a certain rate of success. So just, if you apply 10 times, you might get your grant funded only a small number of times, maybe 2 or 3 times might already be good, right? And so if you're not used to this can be quite demoralizing, because you think, well, I need money to do my cool research, but no one wants to give me money. So I guess the way of dealing with that is you sort of accept it as a process, that you just have to send out basically applications, and statistically some of them might get funded. A lot of them might not, but some of them will, right? So if you kind of accept that system, then you're not going to be, then that's not going to be so bad. You're not going to be so upset by it. If you see each grant rejection as a sort of failure and a reflection on you as a scientist, then that's very bad, because then you become demoralized very quickly. So it's a way of, so the uncertainty in funding is this, is something which I think it's important to know about just at the beginning, that actually a lot of your time as scientists is spent applying for funding, which you may not get. And it's kind of so I would say that's one of the bad aspects, one of the difficult aspects of being a scientist. And even if you get yourself into that mindset where you think, okay, this is just a process, I'm just going to play the game, as it were. I'll send in 10 grant applications and maybe I'll get one funded. It's still kind of annoying to do that. When you have to sit down and do it, you think, well, do I really have to do this? So it's definitely not the most motivating thing to do. So you basically have to organize your time, to do that, but also not forget about the other aspects of the job. The second thing, which is slightly more a feature of the science itself, is there is uncertainty in the science, right? By which I mean, when you're doing a research project, you spend most of the time not really understanding. The understanding might come at the very end, because you typically don't do research on stuff you already know about. So you're researching some unknown aspect of your system, and so when you go to the lab, you do some experiments, you get data, and it doesn't make sense, and you kind of scratch your head and you try to sort of work on it, and eventually you get something some idea, hypothesis, which sort of seems to make sense, and you do some more experiments, and it still doesn't make sense, and you sort of kind of have to tweak your idea. This is actually a way, I think this is a common feature of research, and some people love it. It's basically, they see it as a challenge. It's a puzzle solving. It's like working on a puzzle. You don't see the full picture until the very end, and you kind of have to struggle to put the pieces together. But sometimes, but if you know this and you like it, that's great. I came actually from more of a background, or just personally, where I quite liked knowing about things. And this is interesting because a lot of people study science because they want to know about things, right? You want to go to you want to do your degree to find out more about this thing. And the knowledge comes to you. When you study science, the knowledge is kind of secure. So you learn specific things. But then once you finish studying and you start doing research, the knowledge doesn't come easily anymore. You still want to know it, but it takes a lot more effort to know the new stuff. And so, and also there's no certainty that you will get to an answer, and there's no certainty that you'll figure it out. So that was a big change for me, going from being a student and learning to starting to do research. And it was something I found difficult at first. I think I still kind of find difficult at the moment, which is why a lot of my research in the lab, we spend a lot of time validating things and making sure that we really understand what we think we understand, which I think is important, but it's actually, some labs don't spend so much time doing this. And this is, for me, it's just trying to be more certain about what I do know. So it actually affects me as a researcher in how I do my own research. But I think, this uncertainty in science is something which you sort of have to bear in mind. And if you're not ready for it, it can take you a little bit by surprise.
Right. And that second point is something that sort of feel like it's out there, but it's hidden, and we've never had it really talked about. But it's a very, very interesting point.
Right. Yeah.
Now, next question: would you say it's sort of important to pick, given your field as well, to pick a frontier or sort of emerging field to study at, let's say, our age, people who are getting to undergraduates and will probably be doing their own research in 7-8 years, to ensure the fields are still sort of relevant and meaningful after a postdoc? Or do you think maybe it's the personal interest that should be, sometimes they can come as a Venn diagram, as an intersection, but maybe should it be valued more than our interest sometimes, or not?
Yeah, this is another one of those kind of utilitarian questions, I guess, because I guess it depends. Because, yes, so there are benefits to studying an emerging field, which is that when you come to do research in an emerging field, there's probably more money available. You might be able to get funding more easily. And if you catch a field just as it's emerging, you could be like a big pioneer in the field, right? Because typically when fields emerge, there are lots of maybe easy first questions to ask. So you are set to potentially make a really big impact on the field. So from a utilitarian point of view, from a practical point of view, really the best field you can study is one which is on the cusp of being emerging. If it's emerged for too long, maybe synthetic biology is kind of maturing at the moment, but if it's emerged for a while, then the remaining questions are all difficult. So in order to make big progress, you have to work a lot, and maybe you need resources which might not be available to you when you're just starting off. So the difficulty of questions in the field get sort of more and more as the field matures. And when a field is absolutely mature, all the remaining questions are difficult. So it's unlikely that you'll actually make a big impact. But if it's a mature but important field, there's still likely to be money there. So you can probably still get funding. As to what you want to study, you have to balance that with what you're interested in, of course. If you're not interested in the emerging field, then don't study it, because as I said earlier, in the end you want to work on something you're interested in, not just for the practical reason that maybe you can get more money, and of course, again, there's no right answer to this, right? You can choose to work on a really interesting field for you, which is entirely obscure and no one will ever fund, and you can have a great time studying it in uni and then maybe not go into a research career in that field. So, it depends what you'd like to do. And of course, the other thing is it's possible to change fields. We haven't discussed that yet. It is possible to change. I guess I change fields slightly. It is possible to pivot slightly. If you do your undergrad in one subject and you use some aspect of that to do your PhD, and then you use something you learned in your PhD as to start your postdoc, that's also possible.
Right.
As we get to the end of our questions, one thing that we like to ask everyone is what advice they would have for young people. So this can be about a career in science, just be about life in general. So what advice would you give some young folks like us?
Yeah. Find what. Yeah, it's hard. It's actually quite difficult because
putting you on the spot.
I would say again, I'll reiterate this, find out what you really want. Find out what really motivates you, and see how that fits in. You've made a decision. Let's say you've made a decision to study science, but that decision can be motivated by so many different things, right? As I said, it could be motivated by a particular interest, a particular topic you're interested in. It could be motivated just because you want to learn more about stuff. Some people are motivated, to teach, to learn more and then teach other people. And of course, some people are motivated by these external things. You're really good at doing science exams. You can get that first-class degree. You're motivated by the external awards, not actually by the material itself. And these are kind of hard questions to ask yourself because it might not be clear, and you might spend a lot of time thinking about this as you progress through your career. So it's just this idea of really figuring out, really trying to know yourself, really trying to know what it is that drives you. Because when you know that, it helps you make decisions in the future more easily. Because you can see, you can say, well, I have the opportunity, let's say I have an opportunity to lead a lab. I've been given this, offer, this career offer. Do I really want to take it? Does that align with my actual motivation? I think if you can do that, the people who are successful in doing that, I think, will just have a better time in life. Because we do so many things for the wrong reasons, right? Or sometimes we don't even think about the reasons, we just do stuff. And then we're kind of wondering why we're doing these things. So I think that's my main advice.
Sure.
Yeah.
Great. And as our last question, we've planned to sort of make this book of quotes, maybe one or two sentences from each scientist that we have a conversation with. Very short, it can be, again, about life or science in general, but we'd like to have a collection of all of these in the end so we can maybe summarize all these conversations. So if you would like to add a sentence or two to that, that'd be great.
Well, I don't know. This is getting these short sentences are actually much harder to come by. Than you have to think about them for a long time. If I had to say something, it would probably be along these lines. The self-motivation. So, ask yourself, why do you want to do these things? Ask yourself, why do you want to do science? Or ask yourself, why do you want to do whatever it is you want to do? If that can be turned into a short sentence, I don't know.
Yeah.
But I think that's probably the takeaway from here.
Sure.
Great. Thank you very much for conversing with us.
Great. No, it was a pleasure.
Understand.
Thank you very much for the invitation to come and speak with you guys. So I think what you're doing is really wonderful. reaching out to different scientists about their careers, and I wish you best of luck in your careers and in your lives.
Thank you.
Likewise. Thank you. All right. Have a good day and
Thank
see you
you.
later. Thank you. Goodbye.
Sure. Bye bye.
Bye.